Amines are vital organic compounds categorized by the number of carbon groups attached to nitrogen. While they look similar, their chemical reactivity differs significantly. Today, we will explore how to identify them using a specific reagent, nitrous acid. Nitrous acid is unstable, so it is prepared fresh in the test tube by reacting sodium nitrite with hydrochloric acid. This reaction must be kept ice-cold, between 0 and 5 degrees Celsius, to prevent the decomposition of the reagents. The primary goal of this test is to observe physical changes. Depending on the class of the amine, we will see gas evolution, the formation of an insoluble oily layer, or a clear soluble salt formation. Let's look at the mechanisms. Primary aliphatic amines react with nitrous acid to form highly unstable diazonium salts. These decompose immediately, even at low temperatures, releasing nitrogen gas which appears as distinct effervescence, while leaving behind an alcohol in the solution. Primary aromatic amines, like aniline, behave differently. At low temperatures, they form stable arenidiazonium salts. These do not release gas immediately but are crucial intermediates for creating colorful dyes through further coupling reactions. Secondary amines, both aliphatic and aromatic, react with nitrous acid to form N-nitrose amines. These are insoluble in water and appear as a characteristic yellow oily layer. This visual change is a definitive test for secondary amines. Tertiary aliphatic amines simply dissolve in the cold acid to form soluble nitrite salts. No gas is evolved, and no oil is formed, resulting in a clear, colorless solution that remains stable in the cold environment. Tertiary aromatic amines undergo electrophilic substitution. The nitroso group attaches to the para position of the benzene ring, often producing distinctively colored compounds, such as green crystalline solids, which distinguishes them from their aliphatic counterparts. Let's apply our knowledge. If we add nitrous acid to ethylamine, we see immediate bubbling. This gas is nitrogen, confirming that ethylamine is a primary aliphatic amine. The remaining liquid in the tube is ethanol. Next, consider diethylamine. Upon adding the cold reagent, no bubbles appear. Instead, a yellow, oily substance separates from the water. This confirms the presence of a secondary amine through the formation of an N-nitroso compound. When testing aniline, a primary aromatic amine, we see no gas at 0 degrees. However, if we add an alkaline solution of beta-naphthol, a brilliant orange-red precipitate forms. This dye test confirms the primary aromatic structure. If a sample is added to the nitrous acid mixture and simply dissolves without any visible gas or oil formation, it is likely a tertiary aliphatic amine. The amine has simply formed a soluble salt with the acid. Safety is paramount. N-nitrose amines, the yellow oils formed from secondary amines, are known carcinogens. Additionally, diazonium salts can be explosive if allowed to dry. Always perform these tests in a well-ventilated fume hood with proper protection. To summarize, primary aliphatic amines produce nitrogen gas. Primary aromatic amines form diazonium salts that turn into dyes. All secondary amines form yellow oils, while tertiary aliphatic amines form clear soluble salts. Remember the conditions and the visual cues. The temperature control is the most critical factor for success in this identification. These simple observations allow chemists to quickly categorize unknown amine samples in the laboratory. Test your knowledge: which of these would produce a yellow oily layer when reacted with nitrous acid? If you chose dimethylamine, you are correct, as it is a secondary amine. Understanding the chemical behavior of amines through the nitrous acid test is a fundamental skill in organic chemistry. Thank you for joining this lesson. Generated by Vision Solve AI.